Greetings everyone, my name is Nathan. I'm supervised by Dr. Milton Takanishiyama Jr. on Butantan Institute, and I'm going to present our work on cross-study meta-analysis improvements for breast cancer through a web interface. Breast cancer is the most diagnosed cancer among women in the world, and it's usually classified in 4 subtypes: luminal A, luminal B, HER2, and basal-like, being basal-like the most aggressive and lethal subtype and with the worst prognosis. We developed this project because breast cancer continues to be a large health issue worldwide, and thus it's necessary to identify possible new biomarkers to make treatment more effective and cheaper. Together with this, due to larger heterogeneity between samples, it remains a challenge to properly integrate transcriptomic expression profile data. And as gene expression data is widely available at large amounts, meta-analysis is an attractive approach to integrate transcriptomic data and search for possible new biomarkers. These problems remain unsolved, and thus we developed a modular Web 2 in Shine with an intuitive interface to facilitate meta-analysis of breast cancer samples based on gene expression profile. This is the interface of the Web 2. The user can upload own data as reference or use between data from the Cancer Genome Atlas, and it's also possible to upload specific data to be integrated into the meta-analysis and compared with multiple studies. In order to compare studies in multiple conditions, it is necessary to divide samples in groups, and this can be done in 2 ways. The first one is using AIMS classifier that classifies samples on the 4 traditional subtypes: luminal A, luminal B, HER2, basal-like, and normal samples. The other approach is using hierarchical clustering that groups samples with a similar gene expression profile. This approach is useful when working with unknown samples with specific treatments or conditions. An important step in most analysis is calculating the gene fold change, and that can be done with either LGR or the ECCB. Further, the user can select the normalization method used among the 3-minute mean of ME values, relative log expression, and upper quantity. The user can also define an appropriate threshold to select the differential expressed genes for the subsequent analysis. And the environment also allows the analysis to run on multi-core processing. To obtain information about sign-up pathways from gene expression data, enrichment analysis is a useful approach. Pathway enrichment analysis can be made using 3 different databases among Reactome, KEGG, or Gene Ontology, and the enrichment approach can also be qualitative or quantitative. The qualitative approach uses the names of the differentially expressed genes detected on the previous step, while the quantitative approach that uses fast gene set enrichment analysis uses the gene fold change to enrich the pathways. As said before, meta-analysis is a useful approach to analyze large data sets. The user can select a meta-analysis method among rank product, Stauffer, and Fisher, and can also select a p-value quantity cutoff for considering the relevant genes or pathways. The meta-analysis approach will calculate a single p-value for each subtype or study. And after the process is concluded, it is generated an interactive heatmap that on click generates a dynamic table showing the most similar genes or pathways on the pair of studies, allowing further inquiry of the association between studies. You can also see the p-values calculated for each study. And to make it easier to interpret data, some approaches were adopted, such as Venn diagrams, that allows to identify common and exclusive genes and pathways in a group of studies. Here, you can select a group of studies to see more detailed information. The first information is which genes and pathways participate in each group of studies. And to facilitate the understanding of the relationships among studies, the second information represented is the now interactions between genes and pathways for each selected group, and this allows the identification of exclusive or common pathways and associations between groups of studies. The conclusions are that we developed a user-friendly Web 2 providing new insights based on the integration of gene expression from breast cancer samples on multiple conditions. The meta-analysis approaches were efficient to detect non-biological associations, such as a strong correlation between HER2 and basal-like, and important pathways in tumorigenesis like extracellular matrix organization. The similarity matrix results based on the meta-analysis approach were efficient to identify common responsive genes or pathways for a group of studies, and the multiprocessing environment improvement contributed to a much faster runtime on large datasets. Also, the Venn diagrams, graphs, and tabular representations helped explain the associations among group of studies, making it easy to interpret results. And that's it. Thanks!